quickly, what is going on in interphase? What are the three parts of interphase? G1, which means what?
Growth.
Growth. What comes after G1?
S.
S. And that S stands for synthesis. What are we synthesizing in S phase? DNA. Do y'all remember that unit that we had whenever we looked at DNA replication? That's where this is happening. What comes after S?
G2.
G2, which is more growth. Okay? Now, where do we spend most of our time? In cell division and interphase. Very good. Okay? All right, let's move forward. So flip to your next paper. It should say prophase. Okay? Yes. All right. So now we have entered, this is M phase, which is mitotic. So is interphase a part of mitosis? No. It's kind of like the appetizer before the entree. All right, so what we see in prophase, and this is what you have in your paper, your nuclear envelope is doing what? Your nuclear envelope is disappearing.
Disintegrating.
It's disappearing. And is our chromatin still, our DNA still in the form of chromatin, where it's loose? No, instead, what is it going to look like? It's going to become coiled, or what's that compact DNA called? Not chromatin. Chromosome, very good. So it's gonna look something like this. Okay? Now, is this the actual number of chromosomes, y'all? No. But why are we drawing it like that?
Just to represent them.
Say it again?
Just to represent them.
Just to represent. Because how many total do we have inside of our cell?
50 something.
Not 50 something. It is how many?
46.
46. Half from mom, half from dad. All right, and then on your paper, is it at the very top? On your paper, you see these structures right here.
Like a circle and a
Spindle
block.
fibers.
Our spindle fibers are starting to form, and we have these structures called centrioles, and we say that they travel to opposite poles. And what does that mean? Like North Pole versus South Pole, okay? Very good, so everybody represent with your hands prophase really quickly. Show it to me. Very good. So what comes after prophase, y'all? Meta.
In the
Okay,
middle.
so meta. Meta is middle. Very good. So go to the metaphase portion of your handout. Do we have a nucleus anymore? No, we don't. Okay. So what we see instead are our sister chromatids are aligning in the middle. Or another way that you might see it is that what's the middle of the earth called? The equator. All right, so on yours, it looks, it just has those lines drawn.
Oh, so we draw that in the middle.
Yeah, just draw your chromosomes in the middle, okay? All right, Gatlin, what is it that you think of whenever you hear metaphase? Middle, very good. Middle. Metaphase, middle.
Yep.
And then here are your spindle. Well, these are your spindle fibers. And what do y'all associate with your spindle fibers?
Lassos.
A lasso. So they're like
Really
these
don't mean it.
microtubules that are being.
Fishing pole.
Yeah, a fishing pole. That's one way you think about it. All right. So really quickly, show me prophase with your hands. Metaphase. Now, what comes next?
Anaphase.
Anaphase. Do y'all need more time to write this down?
No.
Okay, so really quickly, anaphase. Anaphase, you should think of what? Apart or what? Away. So in anaphase, what do we know is happening to our sister chromatids? They're being pulled apart. And why are they being pulled apart, y'all? To make new ones. To make new what?
Cells.
To make new cells. Very good. Okay, so this is where we see our sister chromatids being pulled apart. Okay? Give me a thumbs up whenever you're ready. Thumbs down. Okay, so what is this? Pro.
Meta.
Meta.
No, we
Okay,
got
bring
one
this
more.
paper in the lab?
yeah, I want
We
to.
got daughter cells.
Okay, that's fine. We can look at that whenever we do
No, I'm
our.
just joking, we don't gotta do it.
Telophase, what do you think about when you think of telophase?
Two.
Why are you thinking about two?
Because telephone, like Kevin Gates.
I ain't gonna lie, that was kinda.
Why do you think of Kevin Gates whenever you think of telephone?
Because he says he got two phones.
But what does that have to do with?
Because there's two of them.
That's actually a good point. He probably does have a point.
I'm not following.
Yeah, that's.
Listen, Kevin Gates song, the song is
If
it's
you
called Two
listen to
Phones.
it, you'll
And
understand.
it's like he got two phones.
I got
So and it says about a telephone. Yep.
Jared, how you know that song?
I've listened to this song before.
Alright, y'all.
I know what.
Compose
what
yourself,
the
compose
lyrics
yourself.
are.
Guys, if we are talking about animal cells, thank you, Gatlin. What is this structure right here called? A cleavage
Furrow.
furrow. Very good. But what is that cleavage furrow? Get it back together. What is that cleavage furrow indicating? What is it starting to look like?
Separation.
Yeah. So you have the formation of your two cells. What else is forming, y'all? Our nucleus is starting to reappear. So this is the formation of our nucleus. And our DNA is not condensed in chromosomes. It's loose now. It's uncoiling. So we have what? Chromatin. So it kind of looks like
All shriveled up.
okay? So this is chromatin.
That looks like them noodles and them little like Lunchable gummy things. Like that you see at the candy store.
Gatlin, did you write down telophase?
Oh, yeah.
Gatlin, do I need to come over there and check your paper? Now, Gatlin, what's wrong with that picture?
It has eggs.
Uh-huh. So you have too much DNA inside of your cell. So what should you probably do again?
Erase half of it.
Okay. All right, and then after telophase, what do we have, Garrett? Garrett is
Cytokinesis.
cytokinesis. On the last paper, you have how many daughter cells?
Two.
Two. All right, I'm gonna go right here, okay? So you have two daughter cells. These are eukaryotic cells, so they have what on the inside?
Nucleus.
A nucleus, very good, okay? Now, this is mitosis, right? So what type of reproduction was this?
Asexual.
Asexual. And in asexual reproduction, what are we producing? Clones or unique cells?
Clones.
Clones. Okay, so just keep it simple, all right? This is what this cell looks like. What about this cell?
Same thing.
Same thing, okay? And
Looks like
how
a
much
Lego.
DNA do we have in these cells right here, y'all? We have a 2N number, which tells us we have a full set, a diploid, okay? Haven, what do you think about whenever you think of dye?
Two.
Thank you, Haven. All right, very good. Any questions on any of this? I want you to get into pairs, and remember, wait, pause. When you go into the lab, I need you to alternate so you can maximize your space. We are gonna cut the yarn up, and I want you to use this paper to guide you as you look at that each stage, okay? All right, let's go.
Pencils in there?
You can cut it.